I am a cell ready to divide. I'm gonna split in half, call it multiply. Through sexual reproduction, the months equal like 9. Us cells can't do that cause we ain't got any time. Pause my seconds, minutes, hours, go to my almighty power.
Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells. Cells with a single set of chromosomes. These haploid cells mature into gametes.
The cell cycle consists of 5 main sections. G0, G1, S, and G2 are all part of interphase. The actual division of the cell occurs in the M phase, mitosis. Mitosis can be divided into 4 stages: prophase, metaphase, anaphase, and telophase. The first stage is called prophase. During prophase, the nuclear envelope, which contains all genetic material for the cell, begins to dissolve. The DNA within the envelope condenses to form chromosomes, as pictured here. Centrioles with radiating spindle fibers migrate to either pole of the cell. The second step is metaphase. During metaphase, homologous chromosomes line up along the middle of the cell. This imaginary wall is often called the metaphase plate. The spindle fibers attach themselves to the middle of each chromosome at the centromere. The centromere provides a sturdy attachment point among the fragile DNA. Here is another depiction of metaphase. Next is anaphase. During anaphase, the chromosomes are pulled apart from each other towards opposite poles via the spindle fibers. This process can be divided into 2 sections: early anaphase and late anaphase. The last step in mitosis is telophase. During telophase, the cell membrane or wall begins to cleave between the separated DNA. At the same time, nuclear envelopes begin to reform. The result of telophase are two genetically identical cells almost completely separated and preparing to go back into interphase. After mitosis is complete, the final product is a cell that is the exact replica of its neighboring cell. It has a complete nuclear envelope formed around the unwound DNA. Let's review the steps, shall we? We have prophase, metaphase, anaphase, and telophase. Look at this chart and see if you can match the pictures with the phase names that we just said. Could you identify them correctly? Hope so.
Let's compare some events in mitosis with those in meiosis I. In prophase of meiosis I, homologous chromosomes synapse, allowing exchange of genetic material. In mitotic prophase, they do not. In metaphase, chromosomes are dragged to the equatorial plate as pairs of homologs in meiosis I and as individuals in mitosis. In anaphase I of meiosis, homologous chromosomes separate. In mitotic anaphase, the chromatids separate.